Hi, my name is Diego Garcia, and I am a third-year physics student from Cal Poly Pomona. Today I will be presenting on my research in the continuum percolation of disks with correlated positions. Percolation theory studies the spanning behavior of networks of objects and how it is dependent on the ratio of those objects to their systems. This ratio is referred to as the percolation threshold. In particular, site percolation studies lattices where the threshold is the ratio of the number of occupied sites to the total number of sites in the system. Then there is continuum percolation, which studies continuous spaces where the threshold is the ratio of the area of one object times the number of objects in the system to the area of the system itself. Here's an example of a system at percolation threshold, in which we can see a spanning cluster was formed, which is shown in red, and all the black clusters do go partway through but do not span. Some applications of percolation theory include semiconductors, studying disease propagation, porous materials, and imaging. The motivation for this research was an article by my advisor on applying percolation theory to the super-resolution imaging of activated fluorophores with overlapping. Two figures from the article are shown on the upper right. In particular, when imaging things like the cytoskeleton, there are likely correlated structures which, if accounted for, could lead to further improved resolution. The 2D continuum percolation of circles has been a solved problem for a while. So what we're doing differently is depositing those circles on previously existing fibers, such that we can correlate those positions of those circles. It is also important to note that our simulated system has wrapping in order to avoid some unwanted edge effects. We have two ways in which we study this system. Monte Carlo simulations via the Newman-Ziff algorithm, which precisely determines the percolation threshold, or we can focus on the subunits of the system, which is my topic in the research group. And there are two ways to extend clusters within the system. The first way is to have fibers that are close enough that may or might not actually bridge, but the circles deposited on the fibers are able to bridge the gap. And the second way is to have enough circles on the fibers such that they can percolate across the fibers themselves and extend the cluster this way as shown in this image. Here's a quick overview of the process we use to analyze the segments. First, we deposit the fibers, then deposit points on those fibers, and find intersections of the fibers, and track the number of intersections and label them, and find the adjacent intersection points. Then I create an adjacency matrix for those points, and then the algorithm goes into a loop where it does one loop of this for each row in the adjacency matrix, because a row in the adjacency matrix corresponds to a intersection point. And then after all that's done, I analyze the resulting data. One important thing to note of the reason why I use the adjacency matrix is because to speed the algorithm up, I am removing connections from points to other points, in which it is a lot faster to set rows or columns in a matrix equal to 0 rather than removing values from a list. Here are some of the preliminary results that we have so far. On the Monte Carlo side, we can see that when the fiber length is around 6 times the diameter of the circle, that there are repeating peaks in the percolation threshold. And on the subunit focus side, here is a graph of the intersection points and the fibers, and then a graph of the cycles that my algorithm found, and a histogram of those polygonal areas found from my algorithm. Future directions for this research will generally be to understand how the percolation threshold depends on the size and density of the fibers. My work specifically will be to implement the internal shapes that I came up with that result from the interaction between the fibers that form polygons and the circles that are deposited on these fibers, where the green lines represent how far the circles can reach into the polygons. I believe that will be important to track this white space that's in between the green lines, specifically because if we go back to a previous slide, we will notice that as we decrease the size of the fibers, then the percolation behavior of the circles will start to take over. And on the other side, if we increase the length of the fibers after a certain point, the percolation behavior of the fibers will start to take over. So if we go back to the polygon, we should be able to see a crossover somewhere along the line as the polygon size decreases between these two behaviors, and that should prove significant. I would like to acknowledge Professor Alex Small, Andrew Froning, Prableen Kaur, and Cecilia Lee for their contributions to the research group as well. Thanks for listening to my presentation.